We just learned how different forces can cause microevolution, a change in the genotypes and phenotypes from one generation to the next. For example, we saw how the Ice Age dramatically reduced genetic variation in the cheetah population. And this type of evolution, known as genetic drift, has actually caused the cheetah population to become endangered. But how do we know that these changes are occurring? How do we know how much genetic variation is in a cheetah population from one generation to the next? How can we prove that evolution is occurring or that a population is in equilibrium? These are important questions to answer because, for example, they could help scientists monitor the cheetah population and help save them from extinction. So a branch of science known as population genetics can help us answer these questions. And in today's slideshow, we're going to see how we can analyze gene pools in order to observe evolution. First of all, what is a gene pool? Well, it's not a pool that you go swimming in. It's simply all the genes in a particular population pooled together. So in this population of animals, if we were to take all of the genes from each of the organisms, all of the genes for a particular trait, and group them together, we would get our gene pool. Note, for the purpose of this slideshow, we're going to be dealing with traits that are very simple, traits that are caused by one gene with two different alleles. So our gene pools are going to be made up of two genes from each individual in the population. So if we know the gene pool, we can calculate allele frequencies. We can figure out what percent of the gene pool is dominant and what percent is recessive. And in order to do this, I need to know two things. How many of the alleles in the pool are recessive? Well, if I count them up, I see 1, 2, 3, 4, 5. Out of a total of how many alleles in the pool? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14. So 5 out of 14 alleles are recessive, giving me a frequency of 36%. I can find the frequency of the dominant allele the same way. 1, 2, 3, 4, 5, 6, 7, 8, 9 alleles are dominant out of a total of 14, gives me 64% frequency. Now I could also calculate the frequency of the genotypes, but there's a slight trick to this. Let's say I want to know the frequency of heterozygotes. Here's one heterozygote, 2, 3. Out of a total of 1, 2, 3, 4, 5, 6, 7 individual organisms. So 3 out of 7 gives me 43% heterozygotes. Note that in order to find the genotype, I take the number out of the total individuals, because you need an entire individual to have a genotype. So if I wanted to know the frequency of homozygous dominant, I've got one organism, 2, 3, out of a total of 7, again 43%. Frequency of homozygous recessive is just 1 out of 7, 14%. Let's try this in a population of mice. First of all, looking at these mice, I need to determine which gene or which allele is recessive and which is dominant. Well, if I look at my heterozygote, which has one of each allele, the phenotype is dark color. So that tells me that the white allele is actually recessive. It only took one of the dark allele to get the dark phenotype. So if I want to know the frequency of the recessive allele, I count up my recessive alleles, 1, 2, 3, 4, 5, 6, 7, 8, out of a total of 2, 4, 6, 8, 10, 12, 14, 16, 18, 20. 8 out of 20 gives me 40% are recessive. Now we can count up the number of dominant alleles. Out of a total of 20 would give us 12. 12 dominant alleles. Or I could do a little bit of subtraction and get the same answer a little bit quicker. If I want to know the frequency of heterozygotes, I count up the number of individuals. 1, 2, 3, 4. Out of the total individuals, there's only 10 mice here, so I have 4 out of 10 gives me 40% of the individuals are heterozygotes. Similarly, homozygous dominant, I've got 1, 2, 3, 4 mice who are homozygous dominant out of a total of 10 in the population, 40% again. And finally, 2 mice are homozygous recessive out of a total of 10 in the population, and I've got 10%. So it gets a little annoying to keep saying the frequency of the dominant allele and the frequency of the recessive allele, so scientists have a couple symbols to keep things short and sweet. If we're talking frequency of recessive allele, the symbol is Q. Frequency of the dominant allele is P. So population genetics is all about knowing your P's and Q's. Let's just practice using these symbols. So here in this population of butterflies, I can see that the tan color is the brown color is the recessive trait. If I want to know Q, the frequency of the recessive allele, I've got 1, 2, 3, 4 out of a total of 8. Q is equal to 50%. P, the dominant allele, 1, 2, 3, 4, is also, in this case, equal to 50%. Let's try it with these beetles. Now, even though there's a lot of green beetles here, the dominant trait is actually brown, and I can tell that because the heterozygotes, who have one green and one brown allele, end up having the brown phenotype. So if I want to know my recessive allele frequency, Q, I'm going to count the number of green genes. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, out of a total of 12 genes. So Q in this case is 83%. P is simple, only 1, 2 out of a total of 12 are dominant, so P is 17%. Keep this in mind, even though the recessive trait takes 2 alleles, it's not necessarily the least frequent in a population. Alright, so now that we can calculate our P's and Q's, the whole point of this was to be able to observe equilibrium or evolution in a population. So here we've got a population of butterflies in generation 1. If we calculate P and Q, we'd find that Q is 12% and P is 88%. In generation 2, we have these butterflies, the offspring, and if we calculate Q and P, we find that Q is now 25% and P is now 75%. Because the frequencies of Q and P have changed from one to the next, we can conclude that evolution has occurred. It didn't result in an entirely new species, but the basic definition of evolution is a change in frequency of alleles, so something happened here, perhaps mutation, perhaps migration, that affected the allele frequencies. Let's look at this population of mice. In our first generation, I've laid out the different alleles, the dark and the light alleles for you. P is 57%. 50% of these alleles are dark, and Q is 43%. 43% of these alleles, the white alleles. Generation 2, we lost a few mice, and we find that P is now equal to 71% and Q is now equal to 29%. Has evolution occurred? Yes, it has. The frequency of alleles in the gene pool has changed. And the evidence would seem to suggest that natural selection was the force at work here. And the dark mice were being selected for from this generation to survive, reproduce, and pass on their genes. One last thing. Where does all this genetic variation come from? We've already learned this, but it doesn't hurt to review. The ultimate source is mutations. However, sexual reproduction also increases genetic variation because it can combine those mutations in new ways. For example, sexual reproduction isn't responsible for the gene for big noses or bushy eyebrows, but it can combine those genes in new ways. This particular child received a big nose from his dad and bushy eyebrows from the mom. No new genes, but a new combination. Perhaps not a very attractive one, but a new combination. So to sum this up, we have, through the process of mutation, different versions of genes. Meiosis is wonderful because it will recombine those genes through independent assortment in new ways. Fertilization will continue the new combinations, and that's how we end up with genetic variation in our species, and that ultimately is the raw material for evolution.